Okay, so good day everyone. So for today, I will be discussing about antibody structure and function. So last week we discussed about antigen. So today we will be discussing about antibody structure and function. So for our lessons outline, we will be, of course, discussing the tetrapeptide structure of immunoglobulins, the light and heavy chains, the three-dimensional structure of antibodies, and characteristics of the specific immunoglobulins. So there are actually 5 of them. And the theories about the antibody diversity, the genes coding for immunoglobulins, and of course, as an application for immunology and serology, we will also be discussing monoclonal antibodies. So these are the overview of our lessons for today. So let us discuss first antibodies. Okay. So antibodies are actually immunoglobulins. Okay. So these are produced, antibodies are produced in response to antigen. And from last week, we surmised that antigen is actually known as antibody generator. Now, chemically, antibodies are actually serum protein. Okay, so serum proteins. So it's a serum proteins, and we know for a fact that during the process of electrophoresis, there are only 2 types of serum proteins. Either that you have albumin, and the other one is globulin. So between albumin and globulins, immunoglobulins are obviously immunoglobulins. Globulin portion siya, globulin fragment. And then, globulin has 3 fragments. We have the alpha, beta, and gamma. So antibodies, to be exact, is actually a gamma globulin. So it is a protein. In fact, antibodies are made up of 82 to 96% polypeptide and about 2 to 14% carbohydrates. So this is the sugar component. Okay, so majority of the antibodies, the chemical substance of immunoglobulins, are made up of proteins, glycoproteins. So there are 5 classes: G, M, A, D, and E. So later on, we will be discussing them. So antibodies are the key element of humoral immune response. So when you say humoral immune response, there are 2 important cells that are involved here. So these are the plasma cells, and the other one is the memory cells. Okay, so plasma cells are actually important for the secretions of antibodies, while the memory cells, of course, will be important for an amnestic response. So they are important for an amnestic response. Okay, so this is the fraction. No? So I told you a while ago that antibodies are serum proteins, or immunoglobulin is an example of immunoglobulin. And this is the electrophoretic pattern. So we know for a fact that electrophoresis is the migration of proteins in a semi-solid medium. Okay, and this migration of proteins are actually based on electrical charge, and the medium should be of optimal pH. Okay, and the speed of migration also depends on the molecular weight. Meaning to say, the heavier the proteins, then the slower the migration is. So as you can see, the lightest of them all is albumin. Kasi nakalayo talaga yung albumin. And then these are the globulin fragments. Okay, so I have the alpha, and then the beta and the gamma. Alpha has two fragments, alpha 1 and alpha 2. And then we have the beta fragment, and then we have the gamma globulins. So gamma globulin, this is where the classification of immunoglobulin is. So our antibodies are made up of gamma globulin. Okay, so yan. So this is a structure of an immunoglobulin. Okay, so if you are able to see a single structure, such as the one that is illustrated here, this is an example of a monomer. So a monomer is made up of a light chain. So this is the light chain. So there are two pairs of light chain, and we also have two pairs of heavy chain. So the light chain and the heavy chains are being linked together by disulfide bond. Okay. The one that you can see here is an example of a pentamer. So obviously, from the word pentamer is made up of 5 monomers. So it's made up of 5 monomers, and they are being linked together by the J chain. Okay, so the word J actually means joining. The one that is not illustrated here is a dimer. Okay, so a dimer. So a dimer is made up of 2 monomers. Okay, so these are examples of immunoglobulin structure. So this is the tetrapeptide structure of immunoglobulins. So in the illustration a while ago, you've noticed that antibodies are made up of 4 polypeptide chains. So we have 2 large heavy chains. So the one that is in dark blue, ito yung tinatawag natin na heavy chains. And then we also have this 2 small light chain. Okay. So they are being held together by non-covalent forces or the disulfide interchain bridges. Okay. So let me take note of the fact that when we say interchain, okay, it is between the heavy and light chain. So this is, sorry. Okay, so when we say interchain, okay, so when we say interchain, we are referring to the bridges between the heavy and light chain. So this is an example of interchain between the heavy and light chain. And then we also have another example of interchain between the heavy and the light chain. Okay. Now in between the two heavy chain, we also have another disulfide bond. Okay. This is what you call the intrachain. Okay? Because when you say intrachain, it is within the heavy chain. This is the heavy chain. This is another pair of heavy chains. So the one that you found here is also the intra-chain bridges. Incidentally, the intra-chain bridges is where you can also find the hinge region. And the hinge region is important because this contributes to the flexibility of the immunoglobulin. So that even if the epitope is located near to one another or near to one another or far from one another, our immunoglobulin can actually adapt because of the flexibility being contributed body structure. The one that you can find here is actually made up of um, two pairs of heavy chain and two pairs of light chain. Okay. Take note also that each of the heavy chain and light chain would also have the constant region and the variable region. Okay. So this is now the variable region. And then this is the constant. The variable region is important because this is where the antigen binding site occurs. So this is the FAB and this another FAB. Okay? So the antigen binding site will it's like it is somehow adaptive to the epitope. So epitope is the antigenic determinant of the antigen. Okay, so that is the fragment for antigen binding. And with that, it gives us the fact that a monomer has two valences because of the fact that we have two FAB region. While a dimer has 4, while a pentamer has 10. Okay. So the immunoglobulin has a single unique variable region. We call it the amino terminal end. Okay. So the variable region is the amino terminal end. Okay. While the constant region is the carboxyl terminal end. So usually the carboxyl group is actually found here. The carboxyl group is found here in the constant region. Now, if we're going to dissect this particular structure, you would notice that antibodies has variable heavy chain. So that is the VH. And then they would also have the constant heavy chain. The constant heavy chain is further divided into 3. Constant heavy chain 1, constant heavy chain 2, and constant heavy chain 3. Okay. Now, if you will be looking at the light chain here has variable region also. We have the variable light chain, and then we have also the constant light chain. Okay. Now, if you will be asking me, a while ago, I introduced to you the FAB region, right? The FAB region is the fragment for antigen binding. So, antibodies, therefore, can be aggregated into three major parts. So, you have the 2 FAB, and then you have the 1 FC. FC, or the fragment crystalline. Okay, so in close-up view, this is the FAB, and then this is the FB, or the fragment crystalline. Okay, so before we discuss this one, let me tell you the fact that the FAB region, therefore, is made up of variable heavy chain. This is the variable heavy chain, and then the variable light chain, and it is made up of constant heavy chain 1 This is the CH1. And it is also made up of constant light chain. This is the constant light chain. So if I ask you, what are the domains found in FAB? We have the variable heavy chain, the variable light chain, the constant heavy chain one, and the constant light chain one. Okay. So there is one enzyme, and that is known as the papain. So papain has the ability to break the immunoglobulin molecules in the Hinge region before the HH interchain disulfide bond. Okay, between the heavy chain interchain disulfide bond. So this results in the formation of two identical fragments. No, so that's the reason why it is the enzyme papain that allows us to identify these particular fragments. Okay, so the fragment crystalline, on the other hand, has no antigen binding ability. So where's the fragment crystalline? Here is the fragment crystalline. Okay, so fragment crystalline has no antigen binding ability. So it represents the carboxyl terminal hubs of two heavy chains. And the fragment crystalline is also being held together by the disulfide bond. Although fragment crystalline, the reason why we call it fragment crystalline is because this is the one that crystallizes upon purification. So it cannot bind with antigen. Though it cannot bind with antigen, it is important in opsonization. So opsonization mediates in phagocytosis. So the process of opsonization mediates in phagocytosis and also in complement fixation. So for example, this is the antigen. Okay, so we have here the FAB. Okay, and then we have here the complement. Okay, then this is the antigen. It is the FC portion that binds with the complement. However, take note that the FC portion of the immunoglobulin can only bind to two classes of immunoglobulin. Okay, so these are IgG and IgM. So the fragment crystallines of immunoglobulins can only bind, okay, to two classes of immunoglobulin, IgG and IgM. Okay, so to dissect more about the FAB fragment, a while ago, I told you that it is made up of one light chain. It is made up of one light chain. So this is the light chain and one half of the heavy chain. And they are being held together by the disulfide bond. So the reason why there are three fragments, because of digestion with the enzyme papain. So the cut is actually at the hinge region. So this is the hinge region. Okay, and you usually indicate FAB2, which means that in a single monomer, there are two valences. Meaning to say, in a single monomer, there are two FABs, fragment antigen binding. And if the papain fragments it into three fragments, there's another enzyme called pepsin. And pepsin, actually, digestion of pepsin will fragment the FC portion into pieces until it crystallizes. That's the reason why it is called fragment crystalline. And that is actually when it is actually being treated with another digestive enzyme or digestive protein known as the pepsin. Hence, fragment crystalline. So the two at the upper portion, we call it FAB, fragment for antigen binding. So this one, fragment crystalline. Incidentally, fragment crystalline is made up of two heavy chains. And to be more exact, it is made up of CH2 and CH3. Okay, so I hope it's clear ha, kasi yung CH1 is found in the FAB. So the FC portion has CH2 and CH3. Okay, constant heavy chain 2, constant heavy chain 3, and in between, you can actually find the carboxyl terminal here. Okay, so this is the structure of the immunoglobulin. So, why do we call it variable region and why do we call it the constant region? It's because of the number of amino acids. Okay, so it says here, the variable light chain has about 110 amino acid as well as the constant light chain. The variable heavy chain would also have 110 amino acid, while the constant heavy chain will have about 330 to 440 amino acids. Okay, now the constant heavy chain determines the class of immunoglobulin. So I will go deeper into this one later on. But take note that it is the constant heavy chain that determines the immunoglobulin classes. Okay. So let's talk about first the light chain. Light chains are known as the Bence-Jones proteins. I hope you already tackled this in your AUBF, Bence-Jones proteins. Nonetheless, L chains are secreted by malignant plasma cells. So it was discovered by Dr. Henry Bence-Jones, particularly in those patients with multiple myeloma. So when we say multiple myeloma, it's actually the cancer of the bone marrow. And the light chains are actually made up of kappa and lambda chain. And combining the two, we have about 200 and 220 amino acids. Okay, so again, it is made up of constant region and variable region. So this is the 3D structure of the light chain. So the ribbon-like 3D structure of the light chain. So as you can see in this particular illustration, B strands. So we call it the B strands. And they are actually characterized as wide regions, while other regions are like the narrow strings. Okay, so each of the two globular domains consists of a barrel-shaped assembly of 7 to 9 anti-parallel B strands. Okay, so there are also 3 hypervariable regions in the 3-dimensional structure of the light chain. So we designate it as CDR1, CDR2, and CDR3. Okay, so this one are flexible loops. Okay, as you can see, they are highly flexible and they are projected outward from the amino acid and terminal of the beta of the variable light chain. Okay, so they are highly flexible because they can actually adapt, can actually bind with the epitope or the antigenic determinant, obviously, of the antigen. Okay, so this is the three-dimensional structure of the light chain. Now, the heavy chain is made up also of variable region and constant region. So the first 110 amino acids in the heavy chains are variable region, while the remaining are the constant regions. Okay? So, katulad ng sinabi ko kanina, it is the constant heavy chain that determines the class of immunoglobulin. So there are actually three regions, okay, of the constant heavy chain. So we have the CH1, which is found in FAB. CH2 and CH3 is found, they are found in the FC portion. Okay, so again, the heavy chain and the light chain. So the heavy chains are unique to each class. So that's the reason why I told you specifically that the constant heavy chain determines the immunoglobulin class. So for example, if gamma chain is present, then we are referring to a particular type of immunoglobulin known as IgG. If mu chain is present in the heavy chain, then the class is determined as IgM. If alpha chain is present in immunoglobulin, then it is designated as IgA. If delta chain is present in the heavy chain, then the immunoglobulin is designated as IgD. If epsilon chain is present in the immunoglobulin, heavy chain immunoglobulin, then that immunoglobulin class is designated as IgE. So as you can see, the heavy chain is unique and it determines the class of immunoglobulin. And with that, we now have the 5 classes of immunoglobulins. Okay, so these are the 5 classes. So heavy chains, morphologically speaking, may also be categorized as isotype, allotype, or idiotype. So these are what? These are the variants or the variations of the heavy chains. Okay, so an isotype is a unique amino acid sequence that is common to all immunoglobulin molecules of a given class in a given species. So that amino acid is present, for example, in the IgG of human. Allotype is a minor variations of asotype sequence that are present in some individuals but not in others. So when we say isotype, all of our amino acids are the same. But in allotype, you may have a different amino acid. I may have a different amino acid sequence. But those differences are just minor. But when you say idiotype, it is the variation in the entire variable regions. Give and this particular idiotype will allow us to have individual antibody molecules specificity. So to give you an idea again, to explain, isotype is common to all of us. So there are certain amino acid sequences that are common to you, common to me, for as long as we're both humans. But if there are minor variations in that particular sequence, that is classified as allotypes. But if it's really unique to myself, and that determines the antibody molecular specificity. Because the variation is seen not just in few sequences, but in the entire variable regions of the immunoglobulins. Hence, that is called as idiotype. So, this is how we illustrate the isotype. So, isotype, we have the same sequence in a certain species. Allotypes are just minor variations. But if the entire variable region is changed, then we call it the idiotype. And idiotype results into specificity. This is the outcome of idiotype. Okay, so I hope the variations in the heavy chains of the antibodies are clear. Isotype, allotype, and idiotype. A while ago, I introduced to you the Hinge region, which is located somewhere here. The Hinge region is the segment of the heavy chain. It is found in between the CH1 and the CH2. Okay, so it's found between the CH1, this is the CH1, and then this is the CH2. Somewhere in between here is the Hinge region. So the most common amino acid found in the Hinge region is called proline. Okay, and it allows flexibility and hydrophobic residues. A while ago, I told you about the importance of flexibility. Why flexibility is important for immunoglobulins? It's because flexibility contributes to the way immunoglobulins operate. So for example, there are two FAB, and one FAB can bind to another antigen, and another FAB can bind to another antigen. So I hope you get it. And it also assists in the initiation of complement cascade. So that's the importance of flexibility. So this is antigen 1, and then this is antigen 2. There are different antigen, but because of the flexibility of the Hinge region, they can operate independently. Okay, so that's the importance of the Hinge region. Now, immunoglobulins, during the introduction, I told you that they are glycoproteins. So the term glyco means that immunoglobulins would also have sugar component. Okay, and it is found in all types of immunoglobulins, meaning to say, across the 5 classes of immunoglobulins, we have a sugar component. However, that particular sugar component is localized between the CH2 of the 2 domains. So again, let us draw again. Okay, so CH1 is here, CH2 is here. So most likely, the sugar component is found here. And this one is very important because if the immunoglobulin is purely protein, it becomes insoluble because the carbohydrate portion of the immunoglobulin increases the solubility. It increases its solubility, and therefore it provides protection against degradation. And because of the sugar component is found at the Fc portion, it also enhances the functional activity of the Fc domains. So that is the carbohydrate portions of the immunoglobulins. Okay? So I think I have already discussed the interchain and in the intrachain disulfide band. So this is another structure of immunoglobulins in 3D. And then this is the 3-dimensional structure, which I have already discussed. The highly flexible loop of CDR1, CDR2, and CDR3. I have already discussed that, so let's skip that one. Okay, from this point, let us now discuss the individual immunoglobulins. Yes, so the first thing that we will be discussing is the IgG. As you can recall, IgG has gamma chain. So it has gamma chain in the heavy chain, and it is the most predominant immunoglobulins in humans. In fact, 75 to 80% of the total serum globulin is made up of IgG. So it has the longest half-life of any immunoglobulin class. So it's about 23 days half-life. Okay. So IgG is also known as the secondary response. So IgG is also known as secondary response because after IgM, the next antibody that would predominate upon exposure to antigen is IgG. So that's why it is called as secondary response. So there are 4 subclasses of IgG. Okay, so we have the IgG1, which predominates for about 66%, followed by IgG2 for about 23%, and then IgG3 is 7%, and the least abundant among them all is the IgG4. So how do we differentiate IgG1 from the rest of the other subclasses? So they differ in the number and position of disulfide bridges between the heavy chain. So for example, the IgG1 has 2 disulfide chains. So IgG1, IgG2 has 1, 2, 3, 4, 4 disulfide chains. The longest of them all is IgG3. So IgG3 has about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So approximately 13 disulfide bridges. IgG4 has 2, but if you will be comparing it with IgG1, the position of this disulfide bridge is also different. Okay, so that's the basis of the subclasses of the IgG. So IgG3 has the largest hinge region. So obviously, have 13 disulfide bridges. It has the largest number of interchain disulfide bonds. Having said that, we consider IgG3 as the most efficient in binding with the complement. So imagine, so it's very large. So complement can bind. So there are more portion for the complement to bind to. And it is induced in response to protein antigens. Okay, IgG2 and IgG4, 2 and IgG4 have shorter hinge segments, and they are poor mediators of complement activations, and they are involved in responses to polysaccharide antigens. Okay, so ano ba yung mga polysaccharide antigens? Majority of bacteria, especially with capsule. So they are good examples of polysaccharide antigen. Okay, so the major function of IgG. IgG is important because it provides immunity for newborn. Because among the 5 classes of immunoglobulin, it is the only one that can cross the placenta. Why? Because IgG has the lowest molecular weight. And having said that, it is the only immunoglobulin that can pass through the placenta. So it provides immunity for the newborn. This type of immunity is known as the naturally acquired passive immunity. And the biggest contributors of this naturally acquired passive immunity are the maternal antibodies. Okay. There are only two immunoglobulins that can fix the complement, and IgG is one of them. So IgG can fix complement as well as IgM. It can coat antigens for enhanced phagocytosis, so that is known as opsonization. IgG can neutralize toxins and viruses, so prayerfully at the end of your booster dose, you'll have now more IgG. And if you have more IgG, then it can neutralize now viruses. So serologically, it can also participate in agglutination and precipitation reaction, but it is not as efficient as IgM. Kasi IgM is hyper multivalent. So though it can also participate in agglutination and precipitation reactions. So remember that all subclasses of IgG can cross the placenta from IgG1 to IgG4. So macrophages, monocytes, and neutrophils have Fc receptors specific to the Fc region of IgG. Meaning to say, kakampi talaga natin ang IgG when it comes to efficiency of phagocytosis. So a high diffusion coefficient allows IgG to enter extravascular spaces more readily than in other types of immunoglobulins. So imagine, so they can go out of the blood vessel. IgG plays a major role in neutralizing toxins and viruses. So to test for the presence of IgG, we can do agglutination and precipitation. But it says here, IgG is better at precipitation than agglutination. Why? Because precipitation involves soluble antigen or antibodies. And IgG somehow will involve small soluble particles which are more easily brought together by relatively small IgG molecule. Very small because I did mention to you a while ago that it has the lowest molecular weight among the 5 classes of immunoglobulins. Okay, so let us now talk about IgM. IgM is also known as the primary response. If IgG is known as the secondary response, IgM is known as the primary response, primarily because it is the first type of immunoglobulin that will be produced in response to an antigen. So during immune response, IgM is the first immunoglobulin to be produced. However, it is not as long-lasting as IgG. So remember, the half-life of IgG is actually 23 days, but the half-life of IgM is 6 days. So it is not as long-lasting as the IgG. So IgM is a macroglobulin, primarily because it is a pentamer. That being said, we would expect it to have the heaviest molecular weight of around 900,000 Daltons, and it accounts for about 5 to 10% of all serum immunoglobulins. During the maturation of B cells, they are monomer. So when B cells are being matured, particular in immature B cells, they are monomer. However, in secretions, they are pentamer. Meaning to say, you'd expect them to have 10 valences. So the pentamer has J chain, which stands for joining. Okay, this is the linkage point for disulfide bonds between 2 adjacent monomers. And since it is a pentamer, we would expect it to have 10 antigen binding sites. So because of that, it has high valency. And it is found mainly in intravascular pool. So talaga sa loob ng blood vessel natin, maraming IgM. However, because of its high molecular weight, it cannot cross the placenta. And as what I've told you before, we call it the primary response because it is the first antibody that occurs during the immune response, particularly among maturing infants. Do you know that when you were born, you do not have IgM. You only have IgG that you inherit from your mother, and that is a maternal antibody. However, during the first 3 months of life, you will now start to produce antibodies And since it start to produce antibodies, the first antibody that will be produced is the IgM. Hence the term primary response. It is synthesized only as long as antigen remains present because memory cells will start synthesizing IgG, but not IgM. IgM will only be synthesized if antigen remains present. Therefore, during antibody testing, for example, COVID antibody testing, there are two, IgM and IgG. If IgM is present and IgG is negative, it means that you have an acute infection because IgM is only synthesized as the antigen remains. Acute infection or even an active infection. However, if during the COVID-19 antibody testing, your IgM is negative, but IgG is positive, then congratulations. Meaning to say, you are now in convalescent, or it is long after your immunization, and you are able to produce antibodies now. Okay, so that's how we interpret the antibody testing. So kapag mataas ang IgM, negative ang IgG, it means that there are still viral proteins present in your body. And because of that, you still have an acute infection or active infection. But if IgM is already negative and IgG is positive, then congratulations. You now have antibodies against that particular virus. Okay, so that is IgM. So again, this is the chart showing the immune response. So as you can see, the first antibody that will be produced is IgM followed by IgG. So let's say that this is your first dose, and then this is your second dose. During your first dose, this is the antibody titer. IgM and IgG, not yet that high. But during the second dose, IgM increases, but eventually it is not as long lasting as the IgG. It will eventually go down, but IgG will go up higher. Okay, so that's the reason why primary response, IgM, secondary response, IgG. And IgG is long-lasting, lalo na pag booster. Sino mga nagpa-booster na? So your IgG will shoot up more during the booster dose. Okay, so that is IgM for you. So in summary, the functions of IgM includes complement fixation, agglutination, opsonization, and toxic neutralization. So incidentally, noticeably, precipitation is absent here because they are not efficient for precipitation because of the high molecular weight. But for agglutination, they are more efficient than IgG because of the increased number of valence. So they have more efficiency when it comes to agglutination. Okay, so let's talk about the third class of immunoglobulin. So this time, let's talk about IgA. So IgA accounts for about 10 to 15% of all circulating immunoglobulins in the serum. And what's so unique about IgA is that there's only one variable region, but three constant regions. And the serum IgA appears as a monomer with a molecular weight of 160 degrees centigrade, 160 degrees, 160,000 Daltons. Sorry for that, no, that's the molecular weight, 160,000 Daltons. Okay, so because of this, we can actually divide IgA into several subclasses. So we have the IgA1, and it is found mainly in serum. So IgA1 acts as an anti-inflammatory agent because it can downregulate IgG-mediated phagocytosis, chemotaxis, bactericidal activity, and cytokine release. So sometimes our immune system exaggeratedly react to the antigen, causing cytokine storm. And because our body has become the war zone or the battle zone, our body will actually take the price. But if you want the inflammation to stop, and then IgA1 will play that part. IgA2 is predominantly found in secretions at the mucosal surfaces. Therefore, IgA2 will contribute to mucosal immunity. IgA1 is a monomer. Okay? IgA1 is a monomer. IgA2 is a dimer. So they are found in the respiratory, urogenital, and even in intestinal mucosa, mucus-associated lymphoid tissues or the Peyer's patches. And it keeps antigen from penetrating farther into the body. Kaya nga, we call it the mucosal immunity. Kaya nga, if the vaccine is delivered through intranasal spray, then IgA will predominate the mucous membrane, and it will prevent the entry of viruses such as COVID-19. So prayerfully, we can have intranasal spray vaccine. So it is more resistant to bacterial proteinases, and they are that are able to cleave IgA1. Do you know that bacterial protease can cleave and destroy IgA1, but IgA2 is more resistant to it. Okay, so IgA therefore is very important. So it is synthesized in plasma cells found in mouth, mucosal associated lymphoid tissue, and it is released in dimeric form. So this is an example of a dimer. So if you notice, if we're going to enlarge this one, dimer would still have the J chain to join the two monomers. Okay, and of course, the epithelial cells will eventually of the mucosal surface will now release the secretory Ige. Okay, so it is captured by secretory component on the epithelial cells. So secretory IgA therefore is very important. So it patrols mucosal surfaces and act as the first line of defense. If you feel like something is actually, if your eyes are irritated and you think that you might have an eye infection, you know the best thing to do, is to cry. Because if you cry, there's a chance that the tears in your lysozyme can destroy the bacteria, and also you stimulate also the secretion of IgA. So that's true, huh? Okay, but the more appropriately explanation for that is the lysozymes in your tears. It's part of the first line of defense. So IgA can neutralize toxins, not because of the lysozyme, it's a different thing from the tears, but the IgA can neutralize the toxins produced by the microorganisms. Those microorganisms that will try to adhere to your mucosal surfaces. And it is possibly transferred can be secreted by the breast milk, by the mammary gland, okay, to newborn in the form of milk during breastfeeding. So that's the reason why breast milk still best for baby, because maternal milk, okay, the breast milk has a lot of IgA, secretory IgA, and that also contributes to a more functional immunity for the neonates. So complexes of IgA and antigen are easily trapped in mucus and eliminated by ciliated epithelial cells of the respiratory and intestinal tract. So that is also one of the reasons why a vaccine such as the CBIN vaccine is being given orally, because poliovirus is an example of enterovirus. They are in the intestine. So if a child receives saving vaccine orally, the antigen will stimulate the Peyer's patches, the mucosa-associated lymphoid tissue, to produce secretory IgE. And aggregation of immune complex may further also trigger the alternate complement pathway. So we will be discussing complement before quiz number 2. Okay, so that is secretory IgA. So neutrophils, monocytes, macrophages could possess receptors for serum and secretory IgA. So they bind to these sites to trigger respiratory burst and the granulation of the cells involved. So both forms IgA can act as opsonin. So be it, so ano, when we say sinabing both forms, either serum or secretory, can act as opsonin or promoters of phagocytosis. Okay, so the fourth class of immunoglobulins that we are discussing for today is the IgD. Extremely scarce in the serum, so made up contributes only about 0.001%. So imagine 2 zeros after the decimal point. So that's how minute the amount of IgD is. It has a molecular weight of about 180,000 Daltons, and it has delta heavy chain. So that's the reason why we call it IgD. And it has a molecular weight of about 62,000 Daltons. So it appears to have an extended hinge region consisting of 58 amino acids. So IgD is more susceptible to proteolysis other than immunoglobulin. So it has a shorter half-life, even shorter than IgM, 1 to 3 days only. However, they say that IgD is important in lymphocyte activation because they are found on the surface of immunocompetent but unstimulated B cells. Okay, so during the development of B cells maturation, the first antibody is IgM. The first antibody that is synthesized is IgM, then followed by IgD. Okay, so take note, this is not during the immune response, but this is during B cell differentiation. So I have actually discussed this one in chapter 4. So it may also play a role in B cell activation and plays a role in regulating B cell maturation and differentiation. So that's the role of IgD. So during B cell differentiation, IgM is produced, then followed by IgD. So it is secreted in the serum, but in the serum, they do not have protective function. So take note that IgD has a function, okay, during B cell differentiation, but not the protective function for our immune system because it does not bind complement. It does not bind to neutrophil or macrophages. It do not cross the placenta. But are they still important? Yes, because without IgD, B cells will not be differentiated properly. And without B cells, we could not have plasma cells. And without plasma cells, we cannot have antibodies. Yes, they are still important. Okay, and the least abundant of them all, count the zeros. We have 1, 2, 3 zeros after the decimal point. This is immunoglobulins, IgE, the least abundant immunoglobulins among the 5 classes. Okay, it has epsilon heavy chain composed of about 550 amino acids that are distributed over 1 variable and 4 constant domains. It is produced by plasma cells found in the lungs and in our skin. And does it ring a bell to you? Why in the lungs and the skin? Because the lungs and the skin are actually the site where type 1 hypersensitivity reaction will take place. otherwise known as allergy. So you can have rashes in their skin. Asthma is also an example of allergy. So I told you that IgE is the least abundant, but the concentration increases up to 10 times during allergic reaction. So pag nagkaroon pala ng allergy, the amount of IgE increases. So it does not participate in complement fixation, agglutination, or opsonization, and it is incapable of crossing the placenta. However, it attaches to basophils, eosinophils, or tissue muscles through the high affinity Fc epsilon Ri receptor. So imagine this is the immune IgE. So the IgE here has the epsilon Ri. The epsilon Ri binds to the mast cell. Okay. Two adjacent AgE molecules on a mast cell binds a specific antigen. The two adjacent IgE molecule is called cross-linkage or cross-link. So for example, this is the mast cell. And this is the epsilon Ri. And then this is another immunoglobulin. And this is another immunoglobulin. If two adjacent molecules cross-links to one another, okay, if two adjacent molecules on the mast cells bind a specific antigen, we call it cross-link. By the way, the antigen here is specifically known as allergen because it stimulates allergic reaction. So cascade of cellular events results in the granulations of mast cells, and the granulation of mast cells will cause the release of vasoactive amines, mostly histamine and heparin. Histamine is the most common preform mediator of allergic reaction. So pag nag-cross-link pala yung IgE natin na nasa mast cells, ma-release yung histamine from the mast cells. And symptoms of allergic reaction will start to occur. Kaya nga umiinom tayo ng antihistamine. Ayan. So, knowing ko pa talaga. So, for example, this is the FCERI. So, yung FCERI will bind the IgE. Ito na nga yung allergen, no? Causing the granulation. Ito yung mga na-release. It could be heparin. It could be histamine. And it results in type 1 hypersensitivity reaction, hay fever, asthma, vomiting, diarrhea, hives, and the life-threatening anaphylactic shock. Life-threatening kasi ang blood pressure goes down. So asthma is in the respiratory cells and the rest are actually in the skin. So doon pala matatagpuan yung IgE. Eosinophil plays a major part in the destruction of large antigens such as parasitic worms that cannot easily be phagocytized. So mahalaga rin ang IgE. So in a nutshell, these are the different classes of immunoglobulins. Okay, so I hope you'll be studying them. And these are the different differentiation so of the 5 classes of immunoglobulin. Noteworthy to mention yung sedimentation rate, which is yung S refers to the Svedberg unit. This is the unit of measurement used when you are using ultracentrifugation. So the one that has the highest ultracentrifugation rate is IgM because of its high molecular weight. So generally speaking, immunoglobulins can do neutralization, immobilization and prevention of adherence, agglutination and precipitation, opsonization, complement activation, and antibody-dependent cellular cytotoxicity. Okay, so you notice that our antibodies are really diverse. And there are several theories that support its diversity, and one of them is the side chain theory, which was established by Paul Ehrlich in the early 1900s. So he postulated that certain cells had specific surface receptor for antigen that were present before contact with antigen occurred. So even without exposure to the antigen, our cells will now have receptors. However, during the time that this antigen is introduced, it combines with the proper receptor to break off and enter the circulation as antibody molecules. So according to Paul Ehrlich, this particular process could be repeated with further contacts with antigens. So the side chain theory is actually not as strong as the theory of the journey and Bernet clonal selection for antibody formation. So I have actually explained this in chapter 4, meaning to say our lymphocytes are genetically pre-programmed to produce one type of immunoglobulin, and a specific antigen finds the particular cells capable of responding to it. So those cells that responds to it, we call it immunocompetent cells. And only cells that have been activated by certain antigen can proliferate. Hence the term clonal selection theory. Okay? So in 1965, Dreyer and Deney, okay, or Bennett, according to them, constant and variable portions of immunoglobulin genes are coded by separate genes. So, and the genes coding for immunoglobulins are found across the different kinds of chromosomes. So chromosomes, as we all know in your genetics, contains building blocks from which genes can be assembled. So our chromosomes, let's say for example, chromosomes number 14 will synthesize the heavy chain. Chromosomes number 2 can synthesize the kappa chain of the light chain. And chromosomes number 22 can synthesize the lambda chains of the light chain. So rearrangement is needed for genes to become functional antibody molecules. So in our immune system, more than one gene controls the synthesis of the immunoglobulin, of a particular immunoglobulin. So for example, heavy chains, particularly the variable region genes, will control the production of the variable heavy, the D and the J. The constant region genes will control the set of C genes, while the L chains lacks a D region kasi nga nasa heavy chain siya. So through a random selection process, these individual segments are joined to commit that lymphocyte to making antibody of a single specificity. So again, these are the genes coding for immunoglobulins. So this is the heavy chain rearrangement until such time that, let's say, for example, the mu chain for IgM is produced. But as you know, notice, no, there are several clusters. So this is the C cluster, this is the J cluster, the D cluster, and the V cluster. They need to be excised. Pag sinabi natin excised, they need to be cut and rearranged in such a manner that it becomes very specific, so that when transcription and splicing occur, the component of mRNA is highly specific for the mu chain to be produced. So it all happens, okay? It all happens uh, in the DNA rearrangement of the genes. The same for light chain. So in the light chain, there is no D cluster, only the V cluster, the J cluster, and then the C cluster. So again, we need to excise unwanted genes and join the other genes so that during the transcription and splicing, only these three genes will be translated and the kappa light chains are produced. Okay. Now we will be discussing about monoclonal antibodies. So when we say mono, it means single. So monoclonal antibodies are actually antibodies derived from a single clone of B cells. Okay, and monoclonal antibodies can be used for diagnostic testing and even for therapeutic purposes. So you know the anti-sera that you are using for blood typing, that is made up of monoclonal antibodies. So in that case, it's being used for diagnostic purposes. But there are some diseases that can also be cured through monoclonal antibodies. So the development is based on knowledge that B cells are genetically pre-programmed to synthesize very specific antibody. And they are derived from a single parent antibody producing cells that has reproduced many times. So that's why I told you it is produced from a single clone of B cells. So I will just show you an animation how monoclonal antibodies are produced because it's quite easier to understand that animation. So I will actually attach the link as well in your Brightspace. But I just want to define some of the terms. So when you say hybridoma, it is a hybrid between a B cell and a myeloma cell coming from a mouse. So when you fuse the B cell and the myeloma cell or a cancer cell from a mouse, that fusion is known as hybridoma. So you will see that in the animation that I will be presenting to you. Okay, so that ends my